A liquid biopsy is a simple and non-invasive alternative to surgical biopsies, which enables doctors to discover a range of information about a tumour through a simple blood sample. This includes searching for cancer cells from a tumour that are circulating in the blood, or for pieces of DNA from tumour cells that are in the blood. It's a research focus for Professor Rik Thompson, who is the Associate Director and Professor of Breast Cancer Research at the Institute of Health and Biomedical Innovation and School of Biomedical Science at Queensland's University of Technology. He is also a Breast Cancer Trials board member. We sat down with Professor Thompson to talk about how liquid biopsy relates to breast cancer treatment and why it's important in metastatic breast cancer.
Yeah, so this relates really to the other end of the spectrum, and so as I said, most of my career has been in late-stage breast cancer or advanced breast cancer or breast cancer spread. And breast cancers use the blood system to escape and move around. They use the lymphatics as well, and there's a sort of an interchange between blood and lymphatics. But so in women with higher amount of breast cancer burden will have more of their breast cancer cells find their way into the blood. And over the years, we've been able to develop techniques to find those needles in the haystack, to find those very small numbers of breast cancer cells in the blood, and increasingly to look for cancer DNA in the blood. And so, because it's sort of like an integrative site, it's a window into the metastatic cascade. It doesn't really matter where it's coming from. So you can get an idea of what's going on all around the body by having a look in the blood. So it's very convenient. It's difficult, technically very challenging, because it's tiny. You might have nearly 1 billion leukocytes in 10 mils of blood, and you might only have 3 or 4 breast cancer cells. And in fact, one breast cancer cell amongst all those leukocytes is a proven prognostic value. So
is it hoped that it would be used more in a disease maintenance way instead of an identification of the first original breast cancer?
Well, the real hope is that it would allow you to tailor therapy, and so these are what we call clinical utility trials. There's many studies with the initial technology, something called cell search technology, that continue to reinforce the prognostic value and the fact that if you've got more cells in the blood, you have a higher risk of relapse, But it was really trying to make use of that and say, well, if I gave a treatment and the cells in the blood went down, but then they went up again, and so I changed the treatment when they went up again, did I have a better survival? Unfortunately, you can see that if the cells in the blood went up again, the patient certainly did worse. So that prognostic relationship's been reinforced by those clinical utility trials. But so far, we haven't been able to see that the patients actually had a better outcome when we did change, when we saw that the therapy wasn't working. And unfortunately, it may well be that we just don't have better drugs. So we're really at a stage with a patient that we're not getting any benefit with the armamentarium that we have. So it's part of the process. And but it's still very useful to be able to know I shouldn't keep giving this drug because it's not actually working anymore. It did for a while, but it's not working. I should change it, and that's what we do. But we're relying less on those clinically overt parameters where, the pains come back or the sizes come back or the other blood markers have come up. We probably can see this earlier, and particularly with the tumour DNA. The tumour DNA is becoming very important in a couple of ways. One is it's easier to see, I suppose. Rather than looking for 1 or 2 cells in 10 mils of blood, you're looking for probably thousands of copies of DNA, and so it's a little bit of an easier target. But it's still, those thousands of copies are hidden amongst billions of copies of DNA of normal cells, which are circulating in the blood, etc., and coming apart. But we're getting much better at predicting or knowing and being able to measure the actual DNA changes that are occurred in a tumour, and I think better technology to be able to find those in that haystack as well. And it has implications for immunotherapy, which has been a wonderful thing in lung cancer and melanoma, and less so in breast cancer. But triple negative breast cancer is more amenable to immunotherapy, and there's some terrific trials going on, lots of terrific trials. And tumour mutational burden is a key issue with how well immunotherapy can work when we can re-excite it or we can use the treatments that we have. And so monitoring the mutational state, at a DNA level in the blood is going to have some great benefits in immunotherapy as well.
That was Breast Cancer Trials Board Member Professor Rik Thompson. If you'd like to find out more about breast cancer trials and our open clinical trials, visit breastcancertrials.org.au. If you'd like to keep up to date with the latest in breast cancer research, open clinical trials, stories and breast cancer trials activity, make sure you subscribe to the Breast Cancer Trials podcast and follow us on social media. If there's a topic you'd like us to cover on the podcast with our team of experts, make sure you get in touch. And if you enjoyed this podcast, make sure you tell those in your support circles to tune in.